Hi friends, welcome to your lesson on the brain. We're going to start off with what structures make up the brainstem, and we're going to work our way through different sections of the brain. So we have the brainstem, the limbic system are the first parts to the lesson. So what structures make up the brainstem? And I've included this diagram in your lesson notes. We're going to go over the thalamus, reticular formation, pons, medulla, and those are the four parts that make up the brainstem. So we're starting in the bottom on the right-hand side in the diagram with the medulla. The medulla is at the base of the brainstem, and this is what controls your heartbeat and breathing. You don't need to write where it is in your notes because you have the diagram. Just write what its function is. The function of the pons, they are just above the medulla. So if you go up, you can see the pons. The pons controls sleep and helps coordinate movements. Moving up from the pons are the functions of the reticular formation, and then that's a nerve network that travels through the brainstem into the thalamus. So if you see that darker red traveling up through the center of the brainstem, that is the thalamus, the reticular formation going into the thalamus. And its purpose is to help control arousal and filters incoming sensory stimuli. And last, at the top of the brainstem is the thalamus, and its main function is for relaying. It's a relaying station for incoming and outgoing sensory information, with the exception of smell. So moving on to the limbic system. So we're starting at looking at the bottom of the diagram. You have where the word hippocampus is, that like light pinkish, whitish, that's the brainstem. So we're moving up from the brainstem. So the top of the brainstem is the thalamus, and we're moving into the limbic system. So we're going to go over the hypothalamus, the amygdala, and the hippocampus. And when we get to class together during the lesson check-in, I'm going to do a cool little hand model for you to help you remember the different parts of the brain using your hand. So we're going to start with the amygdala, and the amygdala are two lima bean-sized neural clusters. So neural clusters meaning bundles of nerve cells. And the amygdala is linked to emotion, fear, and aggression. Good analogy for amygdala is your amygdala is excited when our amygdala is what's responsible for our fight or flight. And so when humans were hunting and gathering and there were fear of predators and tigers and whatever like chasing us when we're hunting for food or gathering for food, the amygdala is what told us like, ooh, something dangerous is nearby, get out of here. So the amygdala is our response to our emotional response with fears and aggression. So what does research suggest about the functions of the amygdala? One study showed that math anxiety is associated with hyperactivity in the right amygdala. You don't need to write any of this down. This is just cool information to know about research relating to amygdala. Another study linked criminal behavior with amygdala dysfunction, and researchers found when people view angry or happy faces, only the angry ones increase activity in the amygdala. So that makes sense because it's all related to emotion and fear and aggression. So it's all these negative concepts. Moving up to the hypothalamus. The hypothalamus is below the thalamus and directs eating and drinking and body temperature and helps govern the endocrine system via the pituitary gland, which is linked to emotion and reward. So I know that part of the endocrine system, we talked about the pituitary gland, and it is part of the brain, but it's also part of the endocrine system. So it's that little yellow part in the brain diagram. And really directing eating, drinking, and body temperature. So the pituitary gland has hormones that help govern the whole endocrine system itself. So what does research suggest about the functions of the hypothalamus? So here we have a diagram of a rat, and the rat is crossing an electrified grid. And on the left-hand side of the diagram, there's a stimulation pedal. So stimulating the reward centers of the hypothalamus will motivate a rat to cross an electrified grid, receiving painful electric shocks in order to reach a bar that will give additional stimulation. So it will go back and forth across that electrified grid, receiving painful shocks in order to receive additional stimulation, which is crazy. And that is because of stimulating the reward center of the hypothalamus. Moving on to the hippocampus. Small structure with two arms essentially that wrap around the thalamus. And so we're looking at the blue part now, the light blue, and you can kind of see those arms or those bands. The hippocampus helps process for storage which are conscious memories of facts and events. So really breaking it down, hippocampus is related to your memory. And a student shared with me a cool way that she had remembered hippocampus, and it's, if there were a hippo on campus, you'd remember it. So hippocampus is all about your memory. And research suggesting about functions of the hippocampus. Humans who lose their hippocampus to surgery or injury also lose their ability to form new memories of facts and events. Those who survive a hippocampal brain tumor in childhood struggle to remember new information in adulthood. And NFL players who experience one or more loss of consciousness concussions may later have a shrunken hippocampus and poor memory. All right, we're moving on to the third chunk of the lesson, which is in the table, which is the cerebral cortex. The cerebral cortex is the intricate fabric of interconnected neural cells and covers the cerebral hemispheres and the body's ultimate control and information processing center. It is divided into 4 regions called lobes. And again, in the lesson check-in, I'll go over with you guys the hand model that helps you remember the different parts of the brain, specifically the cerebral cortex. So the 4 lobes that make up the cerebral cortex, and this diagram is also in your notes, are the frontal lobe, parietal lobe, temporal lobe, and occipital lobe. And they are located in both the right and left hemispheres. Functions of the frontal and parietal lobes. The frontal is involved in speaking, motor movements, judgment, and decision making. And the parietal lobes receive and process sensory input for touch and body position. Temporal. Each lobe receives auditory information primarily from opposite ears. And occipital is vision, receives visual information primarily from opposite visual fields. And so we'll go into this in a little bit, but what that means is, so like what you see in your right eye is received in your left visual field, and vice versa with your what you see in your left eye is perceived in your right visual field. And the same goes for your hearing. So flipping over to the back of your lesson, we're moving into something different. We're going to talk about split brain, and we're going to talk briefly about the tools that are used to study the brain. So we learned on the front about the different parts of the brain, and now we're going to talk about something called split brain. So split brain is the corpus callosum is a wide band of axon fibers connecting the 2 hemispheres of the brain. Okay, a wide band of axon fibers pointed at by that blue arrow, connecting the 2 hemispheres of the brain. The split brain results when the fibers of the corpus callosum are severed, isolating each hemisphere from the other. And I'll show you diagrams of this in the lesson check-in because it's not very clear from this one right here with the blue arrow. So related to the split brain, I need to go into how the eyes are wired to the brain before we get back into more about the split brain. So in each eye, information from the left visual field goes to your right hemisphere of your brain. and information from the right half of your visual field goes to your left hemisphere of the brain. So your left visual field is registered in your right hemisphere and vice versa. So if you look at this diagram, the very bottom, it says visual area of right hemisphere. I'm at the bottom right. Visual area of right hemisphere. So you see the pencil. And what the eye sees in the left visual field is a pencil. What the eye sees in the right visual field is an apple, but that is picked up in the visual area of the left hemisphere. So visual fields. So along the retina of the eye, sense receptors pick up stimuli that's about 2 inches apart, okay? The right sides of both retinas gather information from the left side of what you are looking at and vice versa. So it's that the whole opposite of what you're seeing something in the left, the right part of your brain is picking it up. So getting back to split brain, how is a split brain different from an intact brain? In an individual with an intact brain, information is readily transferred across the corpus callosum from both hemispheres of the brain. But in an individual with a split brain, this cross transference does not occur. Researchers are able to send information to one hemisphere and test patients based on what they can see. So, Michael Gazzaniga studied the split brains of patients. And I'm going to go over in just a minute what he did. And here's an overview of what he did. The first thing, and you don't need to write this down word for word, but just listen to it and then press pause and summarize after I go over the steps. So the first step is patients with a severed corpus callosum, i.e. split brain, corpus callosum means split brain, were asked to look at a dot in the center of a screen. By looking at the dot in the center of the screen, what that did was create a left and right visual field. So the left of the dot, right of the dot. Then the word heart was flashed on the screen. So that the word he was in the left visual field and the word art was in the right visual field. So two words separated by a dot are momentarily projected. The third step is patients were asked to tell Gazzaniga what they had seen. Patients reported seeing the word art. Let's think about it. Due to the brain's cross-wiring, objects in the right visual field are perceived in the left hemisphere. What does the left hemisphere do? The left hemisphere, and this is really important, these are two psychoblurb words and two areas you really need to be familiar with. The left hemisphere contains two association areas, the Broca's area and the Wernicke's area, that are involved in expressive and receptive language. So both of these areas are involved with language, expressive and receptive language. So the patient saw art in the right visual field. That information was processed in the left hemisphere. The left hemisphere controls language, and so the patient said art. Due to the brain's cross-wiring, the left hand, which is controlled by the right hemisphere, pointed to the word flashed in the left visual field. So, recapping, it's important to pay attention to the question in any AP exam, but to make sure that you note whether the term hemisphere is used or visual field is used. So remember, the image in the visual field is processed in the opposite hemisphere. So those are two confusing terms. Visual field is what your eyes are registering, and hemisphere is what is processing what your eyes are seeing. And so what your left eye is seeing is processed in the right hemisphere. What your right eye is seeing is processed in the left hemisphere. And there's typically a question about split brain on the AP exam. So a few differences in function between the left and right hemispheres. Left hemisphere in most people, speaking and language, math calculations, making literal interpretations, controlling the right side of the body. The right hemisphere, perceptual tasks, making inferences, modulating speech, visual perception, recognition of emotion, controlling the left side of the body. So when we're talking about Broca's and Wernicke's areas, association areas, we are definitely speaking about the left hemisphere. And look at the right hemisphere with modulating speech. So we'll also talk about that more in the lesson as well, the check-in. A point to note, very, very important point. Humans do not have two brains. We have one brain with two distinct coordinated hemispheres, the left and the right hemisphere. And humans are not left-brained or right-brained, even though the media will try to tell us otherwise. We use our entire brain to perform countless activities that are integrated. Okay, another exam tip. Notice that authors never refer to your left brain or your right brain, but you have two brain hemispheres, each with its own responsibility. Remember, you only have one brain. So try to stay away from psychology myths when you are studying. All right, last part of the lesson is how do scientists study the brain? So studying the brain, two main ways in this slide is a lesion and stimulation. So with a lesion, brain tissue is destroyed and researchers study the impact on functioning. So taking some brain tissue out of different parts of the brain and then study how that part functions. Also, stimulation. Brain regions are stimulated electrically, chemically, or magnetically, and researchers study the impact on functioning that way as well. So which imaging techniques show structure and which indicate function? So remember, there's different parts of the brain, and then there's how those parts function, or what the function of those parts is in relation to the part and to other parts. So structures are CT or CAT scans, an MRI, and an fMRI. Whereas imaging techniques that show functioning, EEG, MEG, PET scan, and fMRI. So notice that both fMRIs show structures and functions, but nothing else does. I have included for you the chart that details all of this. So there's really not much to write for this, but please pay attention. So EEG, MEG, and CT scans. So EEG is a recording of the waves of electrical activity across the brain's surface measured by electrodes placed on the scalp. MEG measures the magnetic fields. So EEG stands for the E stands for electrodes, whereas the M stands for magnetic. So it's a really good way to remember the difference between those two. And a CAT scan are X-ray photos taken from different angles and combined by a computer to show a slice of the brain structure, and that shows structural damage. A PET scan is a visual display of brain activity that detects where a radioactive glucose goes while the brain performs a given task. It shows activity. An MRI is a technique that uses magnetic fields and radio wave, so magnetic radio, to produce computer-generated images of the brain, and it's way more detailed than a CT scan. And an fMRI is a measure of blood flow and brain activity by comparing successive MRI scans to show brain function as well as structure, and it can show damage. This is the slide that I have included in your notes so that you not only get the name, you get the abbreviation. So you see the name with the abbreviation. So what I said with EEG, electro, magnetic, so magneto. You see the MEG. Computer tomography is a CT or a CAT scan. positron emission, PET scan, magnetic resonance, MRI, functional magnetic resonance, fMRI, how it works, and then examples. So for the example of EEG, symptoms of depression and anxiety correlate with increased activity in the right frontal lobe, and that's a brain area associated with behavioral withdrawal and negative emotion. With an MEG, soldiers with post-traumatic stress disorder compared with those who don't have it show stronger magnetic fields in the visual cortex when they view trauma-related images. It's really interesting stuff. CAT scans, children's brain injuries shown in CAT scans predict impairments in their intelligence and memory processing. Positron PET scans, positron emission, tomography. Monkeys with an anxious temperament have brains that use more glucose in regions related to fear, memory, and expectations of reward and punishment. MRI, people with a history of violence tend to have smaller frontal lobes, especially in regions that aid moral judgment and self-control. And fMRI, years after surviving a near plane crash, passengers who viewed material related to their trauma show greater activation in the brain's fear, memory, and visual centers than when they watched footage related to 9/11 terrorist attacks. So that is really, really intense information about how our brains hold on to stuff and how we process. And of course, the tools used to be able to tell us all of that. So definitely study those 6 tools and understand what they're each for. Last but not least, pay close attention to the chart on the previous slide, like I just said, and check your understanding of the various methods and results of brain imaging techniques. And we will check in on all of this in the lesson check-in.